Hello, it's Mrs. Han. I decided not to use my camera because I have that roll out of bed pandemic look right now, but I wanted to walk you through the cell types lab for those of you who like to get a little help with it. So let's take a look. You can click on this link to get to the Gizmo lab. You might have to log in, but I think I'm just gonna, can I get rid of my picture here? Okay. If you need the login information, it's right here, username, password, and it's called the Cell Types Lab. So the instructions are all right here, but sometimes you just need to see someone else do it. Here's some prior knowledge questions, no wrong answer. It's kind of what your thoughts are. And then you only have to do activities A and B. This is like a 9-page lab, and after looking at it, I was like, this is very cool, but it's like so long. So I did you a little favor and I cut out activities C and D. I made them optional. If you want to go for it, you can. I'll throw in some extra points for you, but you really don't need to because it's a great lab, but it's just so long. It just takes a long time to get through it. So I just wanted you to get the main ideas from it. And so the first thing is you're going to learn about the parts of the microscope. So it says you'll learn the microscope in the landscape. So here's the landscape tab, microscope tab, test for life. It says click on the Elodia leaf. And then it says show all samples if you can't find it, because obviously, how are you going to find the Elodia leaf? But this is it. But if you didn't know that's it, which I didn't know, I would click that and voila, here's Elodia leaf or Elodia. And so it's going to click on it. And then it says switch to the microscope tab. So there I go, switching. And then observe the sample. Okay, by default, the microscope is used at 40x. So this is low power, and then this is medium, and this is higher power. And it says drag the coarse focus slider. So this is the coarse focus, and we're going to first use the coarse focus to get this this into focus. So here, our LED slide is right here, and we're supposedly looking through the top lens. This eyepiece is a lens. And we're going to move this until it comes into focus. Okay, so right there. And then after you do the coarse focus, then you do the fine focus. This is how you use a microscope. This coarse focus will move it in kind of big movements to get it closer, more or less. And then you can kind of do fine adjustment, which will just do tiny movements until it comes into focus. So it's mostly in focus with the coarse under low. And then you can just do a fine adjustment. If it looks blurry to you, it's because I feel like my eyes are just dying. All right. And then it says, what do you see? And here we see these are all these little blocks, but they're really cells. Hope you know that by now. They're cells. And then it says select the 400. So we're going to go, this is medium, and then now we're going to go to large, high focus. And then we're going to just use the fine. When you're on high power, you cannot use the coarse because it's so close. Any coarse movement is too big. So you just do the fine until it's just right where it needs to be. Okay. And then it tells you, oh, they're cells. By the way, if you look in our course page, these are real Elodia cells, and these are the little chloroplasts that are streaming around there, which is very cool. If you see these under a microscope, it's just really nice. They just are very soothing and calming as they float around here. Okay, so then it says, go back to the landscape, click on the human skin, and now we're going to look at the microscope parts. Okay, so let's go back, click on some skin, which sounds really gross, but it is kind of cool. So it starts back at low power. We always start with the coarse adjustment and get it under focus. By the way, if you want to move it up or down or right or left, you can move these things, okay? That's like moving the slide. So then I'm going to go use the fine, make sure it's as clear as possible under low, and then I'm going to go to, I guess it doesn't really say I need to go to high power for this one, but eventually I will. Okay, so it says, what are the similarities and differences? Okay, so we're going to match the parts of the microscope. If you haven't done this before, here we have an eyepiece, okay, and there's a lens up there. It just is a small magnification, like a 10 times what you would see with your eye. Okay, this knob C is the coarse adjustment or coarse focus, and then this tiny knob D is the fine focus or fine adjustment knob. Okay, these lenses right here are the objective lenses. Those are what magnify 40x, 100x, and 400x. So these you can sort of turn on a real microscope to use whichever magnification that you want. And then obviously the slide is going to be down here, and that's labeled F. Okay, so you can fiddle with the focus knobs and see which one is easier to use on low power and which one is easier to use on high power. So if I'm on high power and I move the fine adjustment, okay, that seems to work pretty well. But if I use the coarse focus, it's not that bad, but really on high power, it's the fine focus that you use. And then when you're on low power, the fine focus does almost nothing, but it's the coarse focus that brings it into focus. So coarse is for low power, fine focus is more fine tuning when you get to the higher magnifications. Okay, and then we're going to show the labels. So now it's going to, so we have to get up to high power. When you get on high power and you get it nice and focused, you can show the labels. So here we can see this is one cell, the nucleus is here with the DNA inside, and then this is the cell membrane. And then this kind of liquid filling where all the organelles are going to be floating around, but we can't see them. This is called the cytoplasm. It's kind of like a watery fluid. Okay, and then it says show scale bar. So let's go ahead and look at the scale bar. This tells us how approximately how big these cells are. So if this is 20 micrometers, then we know the length of a cell is a little more, maybe 30, but depending on which way we're looking at the length or the width. But you can sort of approximate, I would say like 30 or 40, 30 wide, maybe 40 long. And if the bar is not quite where you want it to be, you can move this around to get a better look. Okay. So you could say, oh, it's about like 1.5 of these bars. How wide? So how wide is it? Yeah, we would say, I would say maybe 30. Okay, so then you're gonna be looking at the mouse skin. I didn't know if flies had muscles, but I guess they do. And you're gonna list the different organelles in each of these samples, in the cells of each of these. So when you go back, I'm gonna grab our little mouse. skin. Okay, kind of curious what that fly muscle looks like. So you grab it, go to the microscope. It's going to start under low. You have to adjust it with the coarse first, and then you can go to high, and then you're going to adjust the fine focus on high until it's nice and clear. And then you're going to show the labels, which are already there. And so this mouse muscle also has cytoplasm. It has a nucleus. Let's move this out of the way. Okay. It has a nucleus and it has a cell membrane. So these muscles are kind of like long and skinny. If you go to medium power, the cell is like longer than our view. Okay, so these muscle cells are really long, and they have these things called striations, which are these little lines. Okay. But so you would go ahead and list them, say what they all have in common, and it's going to talk about how eukaryotic cells have a nucleus. But so I think all of these are eukaryotic. They're animal and plant cells. They all have nuclei. All right, and then you're going to look at the E. coli, answer those questions. You're going to look at some sand, look at those questions. Okay, and then let's look at activity B. Activity B is just looking at the size. So we kind of already did that, but we'll look at a neuron. Okay, and then we will go to the microscope. We will adjust it with the coarse and the fine. It's focused, and then I will go ahead and move it to 400. Okay, I'm going to move this scale bar for a second. Okay, so this is a neuron. This is a nerve cell, and we've got our brain is all neurons. Okay, we've got cytoplasm, nucleus, cell membrane, and then they have like these long parts coming off are called axons or dendrites. So this is a nerve cell, and we need to give an estimated size. So we always want to give the size in the highest magnification because it's hard to estimate when it's really small. It's hard to kind of see how big that cell is. So I'm going to go back up to high power, and I'm going to see about how big this nerve cell is. And again, it really depends on where you're measuring it, but let's go ahead and do the width. Okay, so if this is 20, then maybe, I don't know, 15 for the cell body. Okay, and then we need to check if it has a nucleus, yes. Cell membrane, yes. Cytoplasm, yes. And special structures would be axon and dendrite. Okay. Number 2, it's saying to click on the nucleus and see if you can write down the functions of the nucleus, cytoplasm, and cell membrane. So if you're not sure what the nucleus does, you can click on that and it will tell you. And if you can't see it, then you can kind of move the stage so that you can read the whole thing. Put that there. Okay. And then for the neuron, it's going to ask you to tell us what the function of the axon is and the dendrite. And compare it to the muscle. Okay, and then you're going to look at a blood sample and compare human and animals, cells. Good. Okay, so that's basically what you need to do for A and B. If you're interested, and I think it's just really cool to see these, so you're welcome to do some of activity C and D. I just didn't want you to feel overwhelmed because it sort of does take some time. But you could look at some of the microalgae, and you could sketch it if you're like, I love sketching cells. Okay, please do. Yeah, especially plant cells, they're just really pretty because they have that green chloroplasts. They have those little green balls, which are chloroplasts. And then you can look at some root hairs, which obviously are going to be underground, so they might not have chloroplasts. Okay, so this is the side of a root hair. These little things coming off are also tiny hairs off the root hair. So you can do that. And then you're going to look at some little creepy crawlies. Again, this is optional, but it's kind of cool. When you have a real microscope, it's just cool to put everything underneath and see what things look like magnified. But here's some protists. And I love looking at, if you ever have a chance to look at like pond water or the ocean water. That's why I don't like going in the ocean, because I see everything that's crawling in a drop of water and I just don't want that on me. But it is very cool to see them when they're not in your bathing suit. So here's like some really cool things moving around. Here's an amoeba. And if you ever read The Far Side, he loves to make cartoons about amoebas. And here's a Paramecium. This is like a one-cell guy. There's like a, what's this? Here's a euglena, which is kind of like half plant, half animal, because look, it's got these little chloroplasts so it can make its own food. And it's got this little flagellum that kind of propels it around. But it's very cool. These are the little guys in the ocean and the pond. And then here, this part is kind of cool because it allows you to test if something is alive or not. So that's going to be the last section where you're going to pick. Actually, I haven't done this one yet, so let's take a look. Let's choose the worm sample. Worm neuron, okay, and we'll test it for life. Okay, we obviously know a worm is alive, and so we're going to test it to see if it makes ATP, if it changes glucose into ATP, which is our energy source. Okay. So when we click play, this means it's positive for ATP. This means it did not produce any ATP, and our sample produced ATP. So that means it is alive. Okay, it did produce ATP from glucose. I guess we can do something that's not alive just to show what would happen. All of those things are alive except for sand and silt. So if we test that for life, we go ahead and push play. We don't have any positive results. The ATP is not produced by the sand and silt. Okay, and then it lets you like turn on and off the light, because some of these samples will produce ATP in the light, and some of them will not, and some of them will produce ATP in the dark, and some will not. So that'll be interesting too. Okay, I hope that makes this a lot easier for you, and that you have some fun looking at the samples. And it's not, a real microscope, but they do a pretty good job making it interesting, making it as close as possible. All right, have fun.